Well, let me say good morning to everyone. It is indeed an honor to be a part of this experience. The conference has sought to intentionally look at the intersection, the interplay, and the integration of science and faith, and the notion of big questions. I pastor a church in Charlotte, happen to serve as chair of the board of trustees of Gordon Conwell, and so we're involved in the formation of men and women who seek to provide service to the Lord's church wherever they find themselves. And both disciplines, whether it's science or faith, are engaged in raising substantive and foundational questions. The problem is that they are often being raised in isolation from each other. And what BioLogos seeks to provide is a context whereby they can be asked in community, considered in fellowship. And one of the areas of significant question and consideration is in the area of bioethics, particularly in the area of embryology and developmental biology. As a believer and pastor, I am informed by the scriptures, which speak of God's intimate knowledge and activity at the microscopic and embryonic level. Consider the words, "For you created my inmost being. You knit me together in my mother's womb. I praise you because I am fearfully and wonderfully made. Your works are wonderful, and I know that right well. My frame was not hidden from you when I was made in the secret place, when I was woven together in the depths of the earth. Your eyes saw my unformed body. All the days ordained for me were written in your book before one of them came to be." What are the implications of this belief for those who conduct embryological research and medical technologies that they are developing? What are some of the bioethical challenges that their research produces? In what ways does the research, does the technological development lead one to affirm and praise God for God's creative work? To help us understand this is Dr. Jeff Hardin, the chair of the Department of Integrative Biology at the University of Wisconsin. In addition to the numerous scientific research articles relating to embryonic development, Jeff is senior author of The World of the Cell. He also received a Master of Divinity degree at the International School of Theology in Southern California, where he met his wife, Susie, who worked in campus ministry with Cru, Campus Crusade for Christ. He is on the National Advisory Board of InterVarsity Christian Fellowship Faculty Ministry and serves as faculty advisor for the Navigators and InterVarsity Graduate Christian Fellowship on the University of Wisconsin-Madison campus. He also serves as a chair of BioLogos. We discussed one big question just before he comes. This is how prepared he is. Who's going to get to the Final Four? Welcome, Dr. Jeff Hardin.
Well, thank you, Claude. Having just watched my badgers go down in flames, I've learned something about suffering through that, I would say. I did my undergraduate work at Michigan State, however, and my postdoctoral work at Duke, so hope is alive. So, yeah, we'll see how that goes. It is great to be with all of you, and I don't think we've had enough of an opportunity as the board of the BioLogos Foundation to do a couple of things. One is to welcome all of you. It is such a treat to see all of you here. This conference is the fulfillment of a lot of hard work and a lot of aspirations. And we are delighted that you're here, and we hope that this has been a blessing to you. The other thing that I would personally like to do on behalf of the board is for all of us to express our thanks to the incredible staff who have made this conference run so smoothly, and to assist all of us in maximizing our experience here. So I wonder if we could just thank all of the wonderful staff at BioLogos. They are terrific, and it is a great privilege to be able to work alongside them. Well, to the topic at hand for today. The title of this talk is Fearfully and Wonderfully Made, Embryos and Ethics. And where are we headed with this? Well, I want to do two main things. I want to talk about embryos. And I want you to get a sense for how amazing embryonic development is. If you were at the aquarium last night, Rick Lindroth talked about wonder, and how wonder is the intersection of three great strands in human society in their search for meaning. And wonder seems to permeate a lot of what we do at BioLogos, and this morning will be no exception there, because I think it's an opportunity for worship. And secondly, we'll talk a little bit about ethics. So we can only scratch the surface here, but understanding embryonic development in a profoundly Christian context is a starting point, I think, for tackling and having perspective on the challenging technologies that seem to be announced on an almost weekly basis. So we'll do both of those things this morning. But let's start with wonder. This is my son Christopher when he was a baby. He's now 30. Yeah, wow. But when you're a parent and you hold that wonderful little baby in your arms, you can't help but be absolutely astonished at how incredibly delicate yet incredibly structured that little baby is. Those cute little fingers and toes, those ears, all of the structures we take for granted. That sense of wonder reflects the processes of embryonic development. And so to the extent that we can understand how embryos are put together a bit better, that can enhance our sense of wonder and our appreciation for the incredible gift that our children are, as Christopher is. And in fact, that's my main goal for my undergraduates. I'm missing an undergraduate course in embryonic development today. I had to podcast the lecture so I could be here. And what I tell my students on the first day of class is that my main goal for them is that they would think embryos are cool. And if they come away from the class thinking embryos are cool, then I've won. That's my main goal for the class. And one of the things that I do on the first day of class is to say that this is not a new phenomenon, people pondering embryonic development. And I read them a piece of Hebrew poetry that Bishop Alexander already read for us, Psalm 139. You formed my inward parts, you wove me in my mother's womb. I'm fearfully and wonderfully made. And that's a cause of thanks for the psalmist. And I talked to the students about how the psalmist is pondering, even though he knows nothing about biological mechanisms, the great mystery and wonder of embryonic development. And I tell them whether they share my worldview as a Christian or not, my goal for them is that they would be filled with a sense of wonder by the end of the class. So that's my goal for all of us this morning. If you think embryos are cool by the end of this, then I've done my job. Now, for me, I get to study embryos on a regular basis. I am like a kid in a very large, very sophisticated candy store, and it is exciting to me to be able to watch embryonic development through the microscope. And in fact, I came to UC Berkeley as a biophysics PhD student. I didn't really know what I wanted to do. I was actually discouraged after I got there. And Roseanne Senchen and I have talked about how I suffered through quantum chemistry, and I hadn't found anything that I really loved until I talked to a professor who was a trainer in the biophysics program, and he said, "Here, look at these embryos." And it was over after that. I was absolutely hooked. And it was one of these embryos. This is a sea urchin embryo. And you can see it's a beautiful embryo. It's simple and yet complicated at the same time. Well, in fact, building an embryo is really a staggering challenge. We all started as a one-celled zygote, that structure in the upper left, if you're looking at the screens. And then things happen, a white arrow here. And then you get something really complicated at the end. It's my son, Christopher. And developmental biologists are interested in what's in that white arrow. That's what we're interested in as scientists. And just to put this in perspective, this is a conservative estimate of total cell number. An infant has 5 trillion cells, but it started as a single cell. We all started as a single-celled zygote. How do you get that incredible richness and complexity? And why does it turn out so well almost all of the time? That's what fascinates developmental biologists. And there's two fundamentally big ideas in developmental biology. One is differentiation. How do parts become different? The ear is different from the finger, from the eye, from any other specialized tissues in the body. How does that happen? And it's not just the parts are different, they have to be constructed into an organized whole. That's the subject of morphogenesis. It comes from a Greek word morphe for form, genesis I think we know. And what we've learned is that differentiation crucially depends on certain regulator proteins that act like switches, genetic switches. They're called transcription factors. When a cell is making these specialized proteins, a whole host of genes and their expression are regulated, sometimes positively, other times negatively. And the result is that different cells express different sets of genes. And one of the exciting things that we've learned over the past several decades is how deeply conserved a lot of these regulatory switches are among all animals. So we learned about a set of these genes called homeotic genes. They control the identity of parts of the body along the head to tail axis of a fruit fly. But it turns out we have 4 such sets of genes that encode proteins that do very similar things in our bodies and when we were embryos. And this reflects a deep evolutionary conservation by which all animals have diversified on this planet. Now, that's not what I study. I study that second thing, morphogenesis. Why do we need morphogenesis? Well, if you've played with Lego blocks, or maybe you have kids who played with Lego blocks and threw them all over the floor, and then you threw them in a bucket, you realize that Lego blocks are specialized. And that pile of Lego blocks on the upper left there, there's differentiated blocks there. But they're not assembled properly. Assembly is required. And that's what morphogenesis is. And that's what my lab studies is how do cells move about? How do they change their connections with one another? And what's the machinery that allows them to do that? So those are the 2 big ideas. And let's then talk briefly about some milestones in human development. So here's a whirlwind tour of human development. We'll look at a couple of at several stages here. And we'll just point out a couple of things. We don't have a lot of time, but the idea here is that we're duking it out with Jennifer Weissman and Deb Harzma for nice media footage because they think only astronomers have that. Not true. All right. Well, as you know, the first big event is fertilization, when a sperm unites with an oocyte or an egg, and this kicks in a dramatic physiological change in the fertilized egg, which becomes something called a zygote. And fertilization itself is impressive in the sense that it's highly contingent. You know, for the average human who arose during using standard biological processes, this is quite a competitive business. A single sperm has a 1 in 250 million chance of being the lucky sperm that unites with that oocyte. So there's tremendous contingency here in where each of us came from, because each of us has a unique genetic makeup based on that particular sperm who united with that particular egg. Well, fertilization creates a zygote. This is what a one-celled human zygote looks like as the genetic material is coming together, those 2 round blobs in the middle of that image. And then the embryo starts to divide, and that process is called cleavage. This is a 4-cell embryo moving down the fallopian tube, the piece of plumbing that transports the embryo after the egg has been fertilized to the uterus where it can implant. And here's a movie that shows how this works. So this is a mouse embryo, but it looks very, very similar to a human embryo. And you can see that cells are dividing. Now there's 4 cells, now 8. They look like a loose cluster of grapes, but then something happens. Oh, they get very tightly compacted. And then by the end of the movie, you can see that the whole embryo fills up with fluid, and that there are some cells at one end of the embryo. And we'll talk about those in a second. The structure after all of this is done is this. This is called a blastocyst. The blastocyst has fluid in the middle, has some thin cells around the outside, and then a clump of cells, which is at the top in this picture. And that's called the inner cell mass. That's what makes the baby. So that's where you came from. And that's where I came from. And the inner cell mass has extremely developmentally flexible cells, as we'll see, that are the key to making a mammal, including humans. Well, after the blastocyst stage, cells separate into layers, and then something dramatic happens. Cells start to crawl around and form something called a gastrula. This is the stage of development that I've spent a lot of my career studying. And in particular, there's a group of cells. You can see a line that goes from top to bottom in this human embryo. That's called a primitive streak. And that's formed by movements as cells move towards the middle and elongate. The movements are complicated, and we understand them best in non-mammals. In particular, my PhD advisor works on frog embryos, and his name is Ray Keller, and this is a movie from his lab. And if we just run this forward, you can see the incredible movements that are going on. All kinds of crazy stuff is happening as cells are migrating, they're rolling into the inside, the whole embryo is getting longer, and the outside is covering over the inner cells in the embryo. Gastrulation creates the body plan of the embryo. We've now been able to image this in mammals. So here's some mouse embryos from Philip Keller's lab at Genelia Farms in Northern Virginia. And what you can see here is that the green and purple cells are different kinds of cells. And if you look in the middle and on the right, there's a group of pink cells. That's the primitive streak forming. And we can watch that form. And we can use computer-aided microscopy to track individual cell movements. So we're getting much more sophisticated at understanding how all of this delicate choreography is working. If you don't think that's cool, there is something wrong with you. Well, after gastrulation, we all look like a burrito. Yeah. This is a Neurulous stage human embryo, and Neurulous stage human embryos roll up, and the central nervous system forms by taking a flat sheet and rolling it in a process called Neurulation. Very dramatic morphogenetic movement, now that you know that word. And here are some salamander embryos doing Neurulation. And you can see that there's a kind of a keyhole-shaped ridge around the outside of the embryo, but that eventually comes to meet on the backside in the middle of the embryo as a sheet rolls up and closes, and this forms the embryonic spinal cord. Dramatic movements, tightly choreographed, carefully coordinated. Well, after that, organ systems begin to form of many types. That simple tube that forms by that folding enlarges in various places to form parts of the brain, and very complicated things happen over a period of months. And that ultimately is what leads to organisms, complicated organisms like you and me. Well, at this point, you may or may not be asking some questions. Now, if you're my wife, Susie, and this is Susie, you would be asking a fundamentally important question. She is the practical theologian in the house, and her question would be, hey, wait a minute, I thought life's a miracle. You've taken all of the mystery out of it for me somehow by studying it in such detail. Well, my wife, there's a point in a way, but I would like to argue that by understanding the detailed mechanisms of embryonic development, we have a deeper appreciation for just how amazing it is. And what we've learned about embryonic development is that the embryo has everything that it needs, given a suitable environment, to unfold in terms of its development. And this is an exciting way of seeing God at work. Charles Kingsley, a 19th century British theologian, said it this way: are we to reverence him, that's God, less or more if we hear that his might is greater, his wisdom deeper than we ever dreamed of? We knew of old that God was so wise that he could make all things, but behold, he's so much wiser than even that he can make all things make themselves. Now, Kingsley was talking about evolution, turns out, but the same thing, this unfolding by God's providence, is occurring during embryonic development. So it's a way of seeing God at work in a new, fresh way. Well, in what ways can we say that God is at work? We've already looked at Psalm 139. God is present. We know that that's true. And somehow, we were skillfully wrought. Now, the psalmist knew nothing about embryonic development at all, except that certain things happen, and women get pregnant, and there's a baby later. I mean, he knew that, but he didn't know anything about early embryonic development. And yet his conviction, if you know anything about the rest of Psalm 139, is that God is everywhere that he has ever been, including before he was born. God was there. God knew him. God was watching him. God loved him. That's the conviction we have as believers musing about embryonic development. And that's why it's fearful, it's amazing and wonderful, but it's also an opportunity for us to praise our great God. Now, we can say something more than that, of course. And that is that our Lord himself made our story his story. We all know the creed. Jesus was conceived by the Holy Spirit, born of the Virgin Mary. Between those two poles is embryonic development. Jesus dignified our development by taking it up into the divine nature. I can't think of anything else that could give more dignity to the study of embryonic development than that. You know, Lucy Shaw put it this way: after the bright beam of Annunciation fused heaven with dark earth, his searing, sharply focused light went out for a while, eclipsed in amniotic gloom. His cool immensity of splendor, his universal grace, small folded in a warm, dim, female space. Jesus knows where we come from because he has traveled the same path. My small group Bible study is studying Hebrews chapter 4 this week. You may know in Hebrews 4 it says that we do not have a high priest who cannot sympathize with us. And the writer of Hebrews goes on to say, he has been tested in every way that we have, yet without sin. Jesus knows all that we have been from our tiniest beginnings as an embryo. Well, I hope that gives you a sense of wonder. Now let's turn briefly, if we can, to talk a little bit about ethics. Let's talk about embryos and ethics. One of the great things about BioLogos, and particularly the theme of this conference, Beyond Conflict, what a relief it is to talk about something beyond conflict, at least for me, is that we have the opportunity to think deeply about the biology of development, but also about the implications of that biology, especially contextualized for our Christian faith. And let me start by a countervailing voice to a Christian perspective. This is Peter Singer, a bioethicist and a provocateur, I think, and he said this: in this respect, experimenting on a human embryo is not to be compared in significance with experimenting on a living, sentient mouse. Singer's doing something here that some thinkers certainly do. He's denying the human difference. And we as Christian believers want to affirm the human difference and talk about what that means for ethics for a few minutes. First, let's consider, does biology have anything to tell us about how we can define that human difference in an ethically meaningful way. Well, there's a problem here, I think, and that is that we've talked about the continuity of development. We all started as that one-celled zygote on the upper left, and then we all end up looking something like my son Christopher on the lower right. But the problem is that that process is a continual unfolding of potentialities that are already present in that one-celled zygote. They haven't all been actualized at the same time, but it's a continual unfolding. So looking for a magic dividing line that will just through dint of the facts help us to decide the value of human embryos is probably going to be difficult. The Ramsey Colloquium a long time ago said this: the embryo is human. Any being that is human is a human being. If it's objected that at 5 or 15 days the embryo does not look like a human being, it must be pointed out that this is precisely what a human being looks like and what each of us looked like at 5 or 15 days of development. And the point is well taken. All of us traversed this trajectory of development. And in fact, one thing we can affirm is that God knows that whole story. And the Bible clearly affirms that children, before they're born, are begotten gifts. That's from Psalm 127. Some of you may have full quivers. I have 2 children, but you may know that song. Not only that, though, the weak and the seemingly insignificant deserve special protection, and this is a theme throughout the entire Old Testament. Not only that, but Psalm 139, which we've already looked at, and you may be familiar with Psalm 51, where David is musing about himself, and the book of Job also, seems to affirm continuity between embryos and more advanced humans. And not only that, but embryos seem to be part of God's plan, at least when calling the prophets. So all of this suggests that embryos matter deeply to God. But let's be honest. Can we say in modern language that the embryo is an image bearer? Can we say the same things about an embryo that we can say about more advanced humans? Well, we need to be honest that the human biblical authors don't know about blastocysts. I mean, they don't. So that's a problem. The Bible is pre-scientific in that way. And so its language doesn't resolve this issue with the certainty that we would like. And as a result of that, sincere Christians, frankly, and Christians in this room I know full well, may disagree about some of these things. I think we can all affirm, though, that passages like the ones we've been discussing may not establish when human life begins. I would actually like to correct that statement in a moment, but they establish God's care and involvement from the very beginning. We know when human life begins. It's just a question of the moral status of the embryo. But I think this encapsulates very well what we can all say about God's deep care for us as embryos. Now, I myself, given the preponderance of the biblical data and the inability of biology to provide absolutely clean dividing lines about watershed moments in development, that causes me to fall back to a more conservative position, and which is articulated really well by a great bioethicist, Lutheran bioethicist, Gil Mailander. He says this: if we're genuinely baffled about how best to describe the moral status of the human subject, we should not go forward in a way that peculiarly combines metaphysical bewilderment and practical certitude by approving even limited use for experimental purposes. So Mailander's saying we should be careful. And I call that the wisdom of reluctance. I think we should be very reluctant not to treat the embryo as an end in him or herself. But we have a balancing act. We desire, I think, as Christians, to use technology to prevent disease. That's beneficence. And we should seek to treat embryos as patients, an end as opposed to a means at all stages of development. And in our modern society, sometimes those first 2 points can potentially come into conflict. We should also be wary as Christians of technological optimism, and that's because of human limitations. You know, Genesis 2 says God placed limitations on human knowledge. That was before the fall. Genesis 3 adds to that the human sinful condition. So we should beware of technological optimism. And the result of all this is that we are in tension as Christians. And that is something that we need to work out together in community. Now, let me talk briefly about two scenarios that will help you get a flavor for how maybe we can think through some of these issues. The first is something called preimplantation genetic diagnosis. And what this involves is removing one cell from an 8-cell uncompacted human embryo. That's the pipette coming in from the right. And it's slurping out one cell. And it turns out the remaining 7/8 of the embryo can survive. And one can genetically assess the removed cell to determine the genotype, the genetic makeup of the embryo. Now, this is possible because embryos like us, which are from a group of animals called deuterostomes, are incredibly resilient. And a guy named Hans Triesch at the end of the 19th century showed this in a killer classic experiment. This is Hans. Here's his experiment. He took sea urchin embryos at the 4-cell stage, and he separated the 4 cells. And to his astonishment, he didn't get 1/4 of an embryo. He got 4 nicely proportioned sea urchin embryos. So somehow changing the conditions radically changed what those cells would become. So there's a resilience built in by changing the conditions. Now, this flummoxed Triesch, and so he was an embryologist, but he became a philosopher. True story, he became a vitalist philosopher, interesting guy. Now, the same thing is true for us, of course. Here are 2 monozygotic twins. They're not identical, but they started as a single embryo that split. So this tells us that human embryos are very malleable as well. Now, we can show this directly with mouse embryos. Here are 3 mouse embryos that have been put together on the left. They produce proteins that allow them to stick to one another, and then they form a single blastocyst. So there's 3 embryos, embryos 1, 2, and 3, but they glom together and you get a single embryo. Wow. In fact, the mouse that you get is the one on the bottom in this picture. So this is really remarkable. So not only can you split cells and make multiple embryos, but you can stick them together to make a single embryo. And the same is true for primates. So this monkey was made by a combination of 6 embryos glommed together. It's remarkable. So this resilience is what allows for this assay to work. And the question is it good or bad? Well, the goal, of course, of this technique is to assess potentially devastating genetic disorders. That is a good motivation that everyone, I think, would agree with. But typically, if the genotype of the embryo that's assessed is incorrect, then the embryo is intentionally destroyed. And that could have huge ethical ramifications. Now, there are more trivial kinds of uses of this technology. It's being used heavily in certain countries, India and China in particular, for sex selection. And I think all of us would reject a use like that. But recognize that the technology itself leads to the intentional destruction of an embryo. Now, there's an alternative, and I would like to seek alternatives to destroying embryos personally. It turns out one can assess a little nubbin, which is a product of the divisions that lead to an egg or an oocyte called a polar body. And you can slurp off that little piece. There's a pipette coming in on the top there, and it's slurping off that little tiny cell. And you can genetically assess that. And if you're really good and you learned your genetics properly when you were in a college undergraduate, you can interpret that data, and you can assess the genotype of the embryo. Other preimplantation genetic diagnostic techniques, as we've seen, don't do that. They take an embryo and remove a cell, and then the decision has to be made whether to destroy the embryo or not. Assessing the oocyte is done before fertilization happens. So there's an alternative, and I would like to seek creative alternatives like that. But there are other issues too. Issues that Christians have to struggle with about the value of humans in our society with differing abilities or different genotypes. And I've thought about this a lot in my own case. My son Christopher, I've shown a lot of his baby pictures. This is Christopher a little bit later. This is from about 5 years ago. He has severe autism, and autism has a strong genetic component. And we thought a lot about what would we have done if there had been a genetic diagnostic technique available to us. And I think Susie and I both would say that Christopher means Christ bearer. He has been a bearer of Christ to us. Because we've seen the love of God worked out in our care for him and in God's care for us, in God's care for our whole family. So we as Christians have a different voice to bring to the table in these kinds of discussions. All right, very quickly, let me talk about one other example, and it's one that's probably well known to you. Remember I said that the inner cell mass, this thing here with the cells on top, the inner cell mass has specialized cells. Well, it turns out that if you isolate these cells, they can make stem cells. And what are stem cells? Stem cells are cells that can divide and produce more cells like themselves, or they can produce daughter cells that can become more specialized. And a particular kind of stem cells called pluripotent stem cells are very special. They can make all of the cell types in your body when you were developing embryo. Remember I said that the inner cell mass makes the baby at the end of the day. You came from your inner cell mass. Well, pluripotent cells, those inner cell mass cells, can make all of those different specialized cell types. So they're very flexible, and that makes them incredibly, potentially anyway, useful for developing therapies, because if we can coax them to make whatever kind of cell type we want, then we could use them to replace tissues that are missing in a patient. The problem is, though, generating these stem cells requires the destruction of an embryo. So the inner cell mass on the cells on the blastocyst on the left side of this diagram are isolated by removing the cells on the outside using an antibody. So you have to destroy the embryo to harvest these cells, and then you grow them in a dish. So making embryonic stem cells involves destroying a human embryo. And destruction of a human embryo intentionally for this purpose has huge moral ramifications. Now, I'm at the University of Wisconsin. Jamie Thompson developed this technology in humans. And in a very thoughtful piece in the New York Times Magazine a number of years ago, he said, if human embryonic stem cell research does not make you at least a little bit uncomfortable, you have not thought about it enough. And I think he's right. So, embryonic stem cells, good or bad? Well, production of new ES cells always involves intentional destruction of embryos. That's a problem. So you'd like an alternative. And more recently, an alternative has become available due to this gentleman, Shinya Yamanaka, who received a Nobel Prize for this work. In 2006, he identified the proteins that are made in these special pluripotent cells that maintain them as very flexible. And what he found was he could force cells to make these proteins, and he could turn them in to these specialized pluripotent cells. And that technology is called Induced Pluripotent Stem Cells, or iPS cells. So he could force cells to make these proteins, 4 of them, and like cellular alchemy, they could become pluripotent cells. And so these are now called Yamanaka factors very commonly. And what you can do then is take those specialized cells and put them in different conditions, and they will differentiate. That's a word we used earlier in the talk. And they can be coaxed to form all kinds of things, heart cells, nerve cells, you name it. Very powerful. Now, iPS cells seem great, and people are working really hard to try to use them in therapeutic applications. They're great because they don't involve the destruction of an embryo. But there are some unintended consequences of technology most of the time, and we talked about that in the first session, didn't we, this morning? And one of those is that one of the cell types you can turn iPS cells into are gametes. So you can take iPS cells and turn them into sperm or eggs. Now, why is that a big deal? Well, we could isolate cells from me and turn them into oocytes. Now, I'm a reasonably normal human male, and yet we could make eggs from my cells. Conversely, a female individual in this room, we could coax to make sperm. Now, the sperm would be carrying an X chromosome, unless we did something really fancy. But nevertheless, these are possibilities that are made possible by the flexibility of these cells. And then finally, they do some weird things when you get groups of these cells together. They can self-organize. And depending on the conditions, they will self-organize in really interesting ways. So one of those examples is shown here. This looks like a slice of brain if you've had any neuroscience in your background, but this is formed from a group of iPS cells in a dish. They self-organize to form what's called an organoid. This one's called a cerebral organoid because it has structures that are associated with your cerebral cortex. And you can make organoids that form all kinds of different tissues. And disturbingly, I think, and it's raising a lot of questions right now, depending on how you set up the conditions of these cells, you can get something that looks an awful lot like an embryo. And those are called gastrulloids. And here are two examples from the Zernica Getz lab and Alfonso Martinez Arias' lab, both in the UK. And these are from mice, but you can use human cells to do this. And in fact, some engineering groups are using human cells, and they're finding that they self-organize in ways that look an awful lot like an embryo. So, for example, here's a comment from an investigator at University of Michigan. We have to be careful using these terms synthetic human embryo because some people are not happy about it. Now, to the credit of this lab, they realized what was happening, and they realized we need to think about what are we creating here? What kinds of properties are present in these gastrulloids relative to a normal embryo? Now, I don't think they're fully functional in a way that requires our moral consideration in this particular case, but these technologies are becoming ever more refined. All right. Well, I hope you've got a sense for the incredible rapidity with which technology is moving, and it's really due basically to discoveries by basic developmental biologists, people like me. And I hope this has been helpful. We can talk more about where this technology might be going in the future in the Q&A. But what are some next steps? Well, be informed. You don't have to be a developmental biologist to understand the basic kinds of things that we've been talking about this morning. Secondly, be critical thinkers. Many things in this area are highly politicized on both sides of the spectrum. Be thoughtful. And perhaps most important in this list, be loving advocates. Because we as Christ followers should be people who embody Christ's love. Finally, though, I hope that you will be wonderful, that you will be full of wonder thinking about embryonic development. I know I am. Hope you are too. Thanks very much.
All right. We want to move into our question and answer time. And we've got several questions that people have submitted by way of our app. And we're going to try to get through as many of these as we can. So Jeff, here's the first question. Genome editing has the potential for one generation to shape future generations in profound ways. They also have the potential to eliminate suffering, such as cystic fibrosis. The question is, are there applications where we should use CRISPR? And for those of us who don't know what CRISPR is, could you explain what that is on human embryos? If not, what makes this application different from the general mission of Western medicine that seeks to alleviate suffering using technology?
Wow, well, I think for those who are going to hear Francis tonight, he'll have something to say about genome editing. Okay. CRISPR-Cas9 is one of a set of technologies that allows us to make specific changes to the building blocks of DNA in our genome. Oh, Francis is actually here, so you could just take over right now. And to do it in a very precise way, so we can hone in on a particular set of sequence of building blocks and change one out and replace it with a different one. And so for certain diseases where the disease is known to be caused by what's called a point mutation, a single building block that is basically a misspelling at that place in the DNA, we could, in theory, not in theory, we could go in and make a change to revert that building block to the right one, so that it's like a spell checker for DNA.
Got it.
So we could, so that technology exists.
Okay.
Now, there are a lot of questions about using it right now because they're what are called off-target effects, although those are unintended edits to other parts of the DNA that you don't know about at the time that you do the one you intend to do.
Right.
Kind of don't want to be doing that. You don't want to be introducing problems that weren't there. So that's kind of a general safety issue. But the issue of editing embryos is, I think, a tricky one right now. There's been a lot of attention paid to a Chinese scientist who late last year announced that he had edited two female embryos, who are now baby girls. And the way that was done, I have a slide at the end of my talk where I quote Francis. We can put it up, but I don't think we'll do that. And, but let's just say that was done in an unimaginably terrible way in terms of lack of ethical responsibility and a number of other things, and that many people in the scientific community are not comfortable with. So, but if we set that aside, in theory, CRISPR does allow you to make a fix, and then that fix can be transmitted to the next generation.
All right. So what advice would you give me as a pastor as I counsel couples considering in vitro, as well as those who have used it but do not think that they could or should implant remaining embryos?
Well, I counseled a couple at the church that my wife and I were attending in Madison who were having trouble getting pregnant.
Right.
And we talked them through this. In the old days, it was very common to do what's called superovulation. So you get a boatload of eggs, and you fertilize all of them, and you freeze the leftover embryos in liquid nitrogen.
Right. So
we have a huge frozen baby problem to this day because of that practice. So there are in vitro fertilization clinics who will only fertilize a total number of embryos that can be all safely implanted. So for me, that's probably a better way to go. It's much more expensive. So I would say that's a kind of a middle ground in terms of in vitro fertilization. I myself feel that that's an appropriate therapeutic intervention for couples that are infertile. But I want to pay very close attention to how are we handling those embryos.
Thanks. In your opinion, at what stage in embryonic development, in your white arrow, right, does it make sense to recognize the embryo as an entity infused with dignity worthy of social protection?
That's a very rich phrasing of the question. I just want to make sure I understand that. So there's dignity.
Right.
Dignity can be inherent, whether there's a social program in place or not. So that's why how I was pondering the question. But I think the intent is, when do we have an entity where laws come in and that entity is under legal protection?
Yes.
Yeah. You know, it's interesting in the United States that we have a complete hodgepodge of laws. So in some cases, embryos are treated as property. So when a couple has frozen embryos, they are treated in many states as property, which is kind of an interesting thing. But if a pregnant woman is injured and loses her baby, or the embryo, the fetus, then that falls under a completely different part of jurisprudence, you know. So we have a lot of inconsistencies in the U.S. about that. You know, I want to affirm that members of the human species are all part of bearing God's image. And so I want to protect all members of the human species. Now that's different from public policy, and Francis and Baldwin Wong is here. He was partly in charge of administering the NIH's policy on embryonic stem cells, for example. They have much more experience in thinking about rational social policies, that they need to hit the sweet spot of the general populace in a way that people can buy into in a majority sense. So I'm not sure what's a rational legal policy there.
So on that slide, there was those single cell and then the arrow that goes to the to your son.
Yeah.
Okay, so for you, it's closer to the one cell.
Yeah, I mean, I want to provide protections for human embryos, sure.
Okay.
But I think in terms of a social policy, that's a different question.
Right. Okay.
So,
Now let's go towards more towards the end of life. The question is, many malformed babies become multi-million-dollar health care recipients within the first few months of life. Just because we can extend life functions, should we? Now, here's the real question. Is there a place for allowing a loving death?
I think absolutely yes. Now, I don't know what's behind the person's phrase loving death. I would want to be very careful about intentionally hastening death.
Right.
Which would be part of maybe a discussion we might have about euthanasia.
Right.
Versus allowing death. I think those are different morally. So I'd want to be careful there. But I don't see anything in the biblical ethos that would lead us to try to extend human life at all costs in all circumstances. I don't see life extension as something that we should be seeking in terms of our physical bodies. So honoring life and treating individuals with dignity that they have by virtue of being image bearers, I think is important. But I don't, I just don't see in Scripture any sort of mandate to try to extend life at all costs. In fact, if we try to do that physically, and that becomes something that sort of becomes idolatrous for us, we've lost sight of the eternal values and the eternal life that God offers us, that transcends our current bodies.
So I was watching Madam Secretary this past Sunday. That's on CBS. And on there, one of the storylines of that episode was the whole notion of vaccination. Fascinating. And so here's a question that has been asked. Some vaccines can trace their origins to fetal cells. Please discuss the ethics of Christians, especially since we may not know the origins of the vaccines.
Francis has actually been in the middle of a difficult discussion about that. Is that fair? Okay, yeah. Because you are right. So there are some vaccines have been produced using fetal tissue, and some of that fetal tissue has come from aborted fetuses.
Yeah.
And so the discussion is, to what extent is one complicit in that initial act when using the tissue, and how should we think about that? And that ethical complicity is very difficult. I had a long talk with Gil Mylander, who I mentioned in my talk, about this, because we are embryonic stem cell central at University of Wisconsin. So I have Christian graduate students who want to know, can I work with what are called stem cell lines, which were generated by destroying embryos and culturing the cells? But I didn't do that initial I didn't
Right.
do the destruction, right?
Right.
So
Right.
to what extent am I morally complicit in that initial act? And the same issue comes into play with the fetal tissue. So, it would be heart-rending to me if we encouraged someone to terminate a pregnancy for this purpose. That would be absolutely horrible. But I think one thing I've appreciated about Francis's discussion is that he's tried to frame the discussion in a way that in which, okay, the destruction of a human life happened. Mm-hmm. Can any good come out of that? Right. And I think that I think in general this would be true for the fetal vaccine situation. But you also, some of you may remember that President G.W. Bush had a very nuanced policy on existing embryonic stem cell lines. So he did not want to provide federal funding for production of new stem cell lines. That changed under President Obama. But existing stem cell lines, you could work with those. Right. And because he felt that, well, some good should come out of something that was a bad, in his view, this was something bad that happened. So, and I thought that was a very nuanced kind of approach. Now, you have to be careful because there's a tremendous pull to maybe be less nuanced about that, I think. So, but Francis, I just want to say I've appreciated your, I just have so much respect for how you've been in the middle of this in public. So I appreciate that. It's an incredibly difficult situation.
Last question. Now, when you said that our story became Jesus' story, you can imagine the questions that were going to come as a result of that one. So here's one.
But I don't have to answer them.
If Jesus went through embryonic development like us, so did God provide a sperm, and was Jesus completely genetically the same as us?
Yeah. I remember reading a paper. So there's the Journal of the American Scientific Affiliation has it's called Perspectives on Science and Christian Faith. That I remember when I was a grad student reading an article, this very convoluted thing involving something called parthenogenetic activation and a translocation from the Y chromosome and all these other things to explain biologically how you could get a male embryo without a sperm. Wow, I was like, really? I mean, I feel the incarnation is a miracle. I mean, I'm totally comfortable saying that. And I'm not aware that we had the technology when Jesus was born to genotype him, so I don't know. Maybe once we invent time travel, we could go back to this. I don't know. But I guess I find those speculations, maybe some people find them interesting. I feel not sure how productive that's going to be.
I'm good with that.
I mean, the hypostatic union is mind-blowing enough without trying to get into biological mechanisms for me. So, yeah.
Join me in thanking Dr. Jeff Hardin.